Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing IIT JAM question paper for the subject Biotechnology conducted in the year 2019. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of IIT JAM Biotechnology, you can go to my channel Omni Bio Entrance. In the playlist select for IIT JAM, you will find the question papers with explanations there. Coming to the first question. The glycosidic linkage in cellulose and amylose are dash respectively. So cellulose is having beta 1,4 glycosidic linkages and amylose is having alpha 1,4 glycosidic linkages. So the answer here is option B, beta 1,4 and alpha 1,4. Then next one. A mutation in the operator locus of the lac operon that confers constitutive expression of beta galactosidase. Now this is an example of cis dominant, because in cis dominant it will affect, that mutation will affect gene expression on the same DNA molecule and not on the other DNA molecule. So operator and beta galactosidase, they belong to the same DNA molecule. Now this mutation, that is the mutation in the operator locus of lac operon, it will prevent the binding of lac repressor to the operator site, thereby resulting in constitutive expression of beta galactosidase. That is cis dominant. Now what is trans dominant? Trans dominant means it affects gene expression on another DNA molecule, not on the same DNA molecule as in case of operator mutation, but in another DNA molecule. Now this mutation, it is brought about with the help of proteins. Now codominant. Codominant means both the alleles have equal effect on the gene expression. That is codominant. And dominant negative means a mutated gene product inhibits the function of the normal gene product. That is dominant negative. Then next one. Which one of the following figures represent the correct sequence of phases in adult eukaryotic cell cycle? So first comes G1, then S, then G2 and M phase. Now if you see A, it is G1, G2, MS. That is not correct. There should be, the S should come in between G1 and G2. So G1, S, G2, M phase in B is correct. C also it is not correct. D also it is not correct. Then next one. At what pH does polygluc in an aqueous solution forms alpha helical structure? Now polygluc, it is a polypeptide and it is composed of glutamic acid residues. Now the alpha helical structure, it is influenced by pH due to ionization of the side chains. So if you see the options here, only one option is being given as 3. The others are 7, 9 and 12. So at what pH does polygluc in aqueous solution forms alpha helical structure? So if you take the consideration of pK of glutamic acid side chain, it is 4. Now 3, if you see, it is less than 4. That is pH, if you take pH as 3, it is less than 4. So most of the side chains will be protonated, or I can say about 50% protonated. And this 50% protonation, it will allow for hydrogen bonding and formation of alpha helical structure. Now if the pH is greater than 4, that is in other options, that is 7, 9 and 12, pH is greater than 4, it will result in deprotonation, and deprotonation, what will happen is it will increase electrostatic repulsion and thereby destabilizing of the alpha helical structure. So 7, 9 and 12 are not the right answers, because it is the pH is greater than 4. Now what will happen if the pH is less than 3? That is just for your information I am telling. Now pH below 3, the side chains are fully protonated. They are fully protonated and thereby reducing hydrogen bonding, and it will result in random coil formation. So at pH 3, polygluc in aqueous solution will form alpha helical structure. Answer is option 3. Then next one. Match the entries in group 1 with the entries in group 2. Nylon. Nylon is adipic acid, P4. Natural rubber is isoprene, Q1. Then starch is a hexose, R2. And myoglobin is an amino acid, S3. So answer here is P4, Q1, R2, S3. Nylon is a polymer of adipic acid, natural rubber of isoprene unit, starch is of glucose which is a hexose, so it is hexose subunits, and myoglobin is made of amino acid, it is a protein, so it is a amino acid subunits. Then next one. The technique which involves impacting samples with electrons is NMR, no. NMR spectroscopy, mainly protons are taken into consideration. ESI, mass spectrometry. ESI is electrospray ionization mass spectrometry. So that is the answer. IR is infrared spectroscopy, and UV visible spectroscopy also doesn't use electrons. Then next one. The evolution of eyes in octopus and humans is an example of. So here it shows convergent evolution. Okay, convergent evolution. They have similar functional traits. That is, they have lens, retina, photoreceptors, but they have different ancestral origin. That means octopuses evolved from mollusks and humans from vertebrates. So there is independent evolutionary pathways. That is example of convergent evolution. What is option A is divergent evolution. What is divergent evolution? Evolution of different traits from a common ancestor. That is divergent evolution. And adaptive radiation is evolution of new species from a single ancestor. That is adaptive radiation. And genetic drift is random changes in gene frequencies over time. That is genetic drift. Then next one. Which one of the following modification occurs both on protein and DNA? ADP ribosylation, it is occurring in protein only, not in DNA. Methylation. Methylation occurs in both DNA and protein. So addition of methyl groups to cytosine or adenine residues in the case of DNA, and in proteins addition of methyl group to lysine or arginine residues. If you take histone modifications, there you can find there is addition of methyl groups to lysine and arginine residues. And histones are proteins. So methylation is the answer. So sumoylation, ubiquitination, all that are found in proteins only, not in DNA. Then next one. Solution of which of the following peptides are prepared separately at a concentration of 1 millimolar? Among these four, which one has the highest A280 ratio? So from among these peptides, you have to see which of the following peptides is having the maximum concentration of tryptophan. Why? Because tryptophan has the highest absorption at 280 nanometers. So if you take A, you can find tryptophan here 1, 2. If you take B, there is no tryptophan. Then C, only one tryptophan. And D, if you see, there is no tryptophan. So maximum tryptophan occurs in A only. So A will be having the highest absorption at 280 nanometers. Then next one. The free energy required to synthesize a mixed anhydride bond of 1,3-bis phosphoglycerate is generated by the oxidation of. So if you remember your glycolysis, you can see that 1,3-bis phosphoglycerate is formed from glyceraldehyde 3-phosphate plus NAD plus PI, giving 1,3-bis phosphoglycerate plus NADH plus H plus, and it is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So here aldehyde is getting converted to acid. So which is the aldehyde? Glyceraldehyde 3-phosphate. It is getting converted to acid 1,3-bis phosphoglycerate. So the free energy required to synthesize a mixed anhydride bond of 1,3-bis phosphoglycerate is generated by the oxidation of an aldehyde to acid. Here NAD is getting converted to NADH plus H plus. So the other options, alcohol to acid, alcohol to aldehyde, is not correct. NADH to NAD plus also is not correct, because in that case energy would be consumed and not generated. So D is also not the right answer. Then next one. Which one of the following parameters change upon doubling the enzyme concentration? Naturally, your Vmax will change upon doubling the enzyme concentration, because Vmax is directly proportional to enzyme concentration. So if you double the enzyme concentration, Vmax also will get doubled. Km, it is independent of enzyme concentration, and it says about the substrate binding affinity. So it is independent of enzyme concentration. Kcat, it measures the turnover rate of the enzyme. And K equilibrium, it is equilibrium constant, and it is independent of enzyme concentration. Then next one. Which one of the following statement is a correct description of the mode of action of Taxol and colchicine? So Taxol, it stabilizes the microtubule, whereas colchicine, it prevents microtubule formation. Okay, that's the action, exact opposite. So D is the answer. Taxol stabilizes microtubules and colchicine prevent microtubule formation. So answer is D. Then next one. In a simple microscope, a lens with negative power is used. No, it is with positive power is used. The focal length of the lens is less than the least distance for clear vision. Least distance for clear vision is your 25 centimeters. So the focal length of the lens is less than least distance for clear vision is the right answer. The focal length of the lens is greater than least distance. No. Magnification depends on the focal length of the lens. Magnification depends on focal length of the lens, least distance for clear vision, and object distance. So these three factors magnification depends, not just on the focal length. So D is also not correct. Answer is option B. Then next one. Which of the following statements is incorrect with respect to bacterial conjugation? It facilitates transfer of genetic material. That is correct. They have asked for the incorrect statement. It requires flagellum. Bacterial conjugation is direct cell-to-cell transfer of genetic material. So conjugation happens through what is called as sex pili. Okay, so it requires flagellum is not correct. That is the incorrect statement, and that is what they have asked in your question. So B is the answer. Then next, if you see C, it can spread antibiotic resistance. Yes. It can transfer virulence factors. That is also correct. Then indole acetic acid is involved in. Indole acetic acid is an auxin. And auxin is involved in, out of these options, gravitropism. Okay, gravitropism. Now what happens in gravitropism is that the auxin accumulate on the lower side of the roots, that is here. It accumulates there, whereas the upper side doesn't have much auxin. So what will happen there is low auxin concentration on the upper side, it will promote cell elongation. Whereas high concentration of auxin on the lower side, it will prevent cell elongation. It will inhibit cell elongation. So what will happen due to this differential? Growth rates will happen, and the upper side it will grow faster than the lower side, and it will bend and direct the, and it will result in curve. This is the curve. So it will result in a curvature, and it will direct the root to grow towards the gravity. So indole acetic acid is involved in gravitropism. Then next one. Match the entries in group 1 with group 2. So bacterial disease over here is tuberculosis, P2. Viral disease is influenza, Q3. Q3. Then protozoa, it causes malaria, R1. And autoantibodies, that is your autoimmune diseases. It is an example given here is myasthenia gravis. So S4. So the answer here is option B, P2, Q3, R1, S4. Then multiple select questions. Pick the correct statements with respect to the interconversion of the topoisomers of a circularly closed double-stranded DNA. So topoisomers, there are two types, that is topoisomerase 1 and topoisomerase 2. Topoisomerase 1, it will cut only single strand of the DNA, and topoisomerase 2, it will cut both the strands of the DNA. Okay, and both of them require ATP for their action. So first one, only one strand needs to be cut. That is correct. Both strands have to be cut. That is also correct. No strands need to be cut. That is incorrect. ATP is required for interconversion. That is also correct. So A, B, D are correct. Then next one. The characteristic oxygen binding profile of hemoglobin shown below arise due to. Now oxygen binding by hemoglobin, it is a sigmoidal curve. It is, you can see over here in the graph. So it shows that high affinity for oxygen is cooperative binding, which means one molecule of oxygen, it will enhance bind to hemoglobin, will enhance the binding of further oxygen molecules to hemoglobin, and that is because hemoglobin is having a quaternary structure. So that is correct. Cooperativity, C is also correct. Subunit dissociation doesn't take place. Conformational change. So there are two conformations of hemoglobin based on oxygen binding. One is the T state and the other is the R state. So T state is the tense state and it has low affinity for oxygen. Whereas R state, it is the relaxed state and it has high affinity for oxygen. So oxygen binding, it will induce conformational change in hemoglobin. So D is also correct. So A, C, D is the answer. Then next one. The advantages of storing chemical energy in the form of starch and not as free glucose is or are that it. Okay. Minimizes diffusion. That's correct. Starch is a large molecule whereas glucose are smaller molecules. So starch will not diffuse very fast whereas glucose will diffuse very easily. So minimizes diffusion. So if you store energy in the form of starch, it will minimize diffusion. So A is correct. Enables compact storage. Again, starch is a polysaccharide. It is bigger in size. Glucose is small, so it will occupy more space. Whereas starch, it will not be able to occupy more space. And because of its large polysaccharide size, it has to be stored in a compact way. So enables compact storage. B is correct. Reduces osmotic pressure. Starch is insoluble and so it will not contribute to osmotic pressure. Whereas glucose, it is soluble and it can result in increase in osmotic pressure if the concentration is high. So reduces osmotic pressure. Yes, starch reduces osmotic pressure. That's correct. Protect against chemical reactivity of aldehyde group. Now starch is having glycosidic glucose linked to by glycosidic linkages to the next glucose. So one glucose molecule is linked to another glucose molecule via a glycosidic bond. So this will protect the aldehyde group from getting reacted or undergoing oxidation in starch. Whereas the glucose it is having a free aldehyde group and it is susceptible to oxidation. So protects against chemical reactivity of aldehyde groups is also correct. So A, B, C and D are correct. Then next one. Which of the following cell types can develop from myeloid lineage? So myeloid lineages, they are the T cells, macrophages and erythrocytes. They develop from the myeloid lineage, and T lymphocyte and B lymphocyte, they develop from the lymphoid lineage. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of IIT JAM Biotechnology being solved, you can go to my channel Omni Bio Entrance. In the playlist, search for IIT JAM and you will find it there. Thank you.